I'm pleased to invite another luminary in the field of cholangiocarcinoma to the stage to speak on our clinical advancements over the last 10 years. Dr. Jennifer Knox is a professor of medicine at the University of Toronto, working as a staff medical oncologist and researcher since 2001. Dr. Knox is currently the head of solid tumor medical oncology group at the Princess Margaret Cancer Center and co-director of the McCain Center for Pancreatic Cancer.
Wow. Good morning, everybody. Indeed, what a pleasure to be here to celebrate the Cholangiocarcinoma Foundation's 10th annual conference. I've been given the task to describe the clinical advances in the last 10 years. And if I know how this works. And I come from Toronto, where you can see we light up the CN Tower green for World Cholangiocarcinoma Day, usually the coldest day of the year in February. So my task is to fill in this number line. Lindsay, who's a hard act to follow, already showed a beautiful timeline, which is nicer than mine. So I was worried about being too long, but my job is done. But maybe you'll humor me. So when I think back on where we were in 2014, well, we know at that point from the study by Juan Valle and others that we had GemCis as a chemotherapy regimen that improved survival. There was a repeat trial in Japan. So this was a standard of care, and a standard of care in clinical research or clinical medicine is important because it's a platform on which you can build. However, what we did not have was prospective adjuvant data, neoadjuvant trials, second-line therapies. Biliary tract cancer needed real attention from pharma and researchers, hence the pay it forward, is that, of the CCF approach. And really, in clinical practice, precision oncology was not a topic of discussion. What did we have in other areas of oncology in 2014? Well, there was this drug, nivolumab. Have you heard of it? It was a first-in-class anti-PD-1 monoclonal antibody that got FDA approval for treatment of melanoma, followed by other solid tumors, quickly really ushering a new era in oncology, but unfortunately not in cholangiocarcinoma. We know the ABCO2 data impact across all the different anatomical subgroups. Other studies, which I won't go into, but important questions at the time, adding an EGFR, VEGF, or MET did not lead to an improvement on this standard of care. So the era of next-generation sequencing began in about 2016 for cholangio. And this was important because we saw publications like this from Melanjavel that basically showed that there were potentially druggable subtypes against across the different anatomical groups. And there were trials underway, basket trials and other things that could perhaps put our patients on. So this was quite eye-opening. Was also followed by important studies such as this one that was looking at the ICGC that really used a multi-omic approach, including epigenetics, et cetera, and showed how heterogeneous this patient population actually is with respect to those omics, but also prognosis. And this really hasn't come into clinical practice too much yet. But as Dr. Bardisi say, there's probably trials planned to try to address some of this. So NGS really launched the genomic-driven studies. It was an area of a time of discovery. We got a windfall. MSI high tumors respond to checkpoint inhibitors. And I think we all remember the first time we saw this slide presented where MSI high cancers responded to pembrolizumab. And you can see cholangiocarcinoma. I remember searching this slide, trying to find it, and there it is in gray with a complete response. So that led to the agnostic indication for DMMR solid tumors, which is important. Now, while all the launch of genomic-driven studies were underway, I'm just going to step aside for a moment and talk about adjuvant therapy, capecitabine, because this is an important area of medicine in which we can improve survival. So we know the Bill Cap trial that looked at capecitabine versus observation after resection, which is an improvement in the median survival, 52 versus 36 months, and this is practice-changing globally. It's been criticized a little bit on its design, and people were hoping to see better, but I think it really needs to be studied or looked at in the context of what else has happened in those 10 years. So for example, the French study gemcitabine oxaliplatin, so a more chemo-intense regimen versus observation, and although it's a smaller, less risky population, it is a negative trial. Bill Cap is positive. The Japanese looked at gemcitabine, negative. They looked at S1, which is another oral fluoropyrimidine. That is a positive trial, practice-changing in Japan. And then more recently, we saw GemCis versus capecitabine in extrahepatic high-risk, negative. That same design is underway in a larger trial in Europe. So there were a lot of lessons learned from this. We did pragmatic all-comer strategy because it's hard to do trials in cholangiocarcinoma, but it really probably diluted out any real signal. Oral 5-FUs remain the winner. There are many lessons, lots written on what we can learn from the Bill Cap trial. I won't go through it all, but I think it points us to considering neoadjuvant as well as radiotherapy approaches and to study more homogeneous populations. There's this recurring lesson in oncology that what works in the advanced setting doesn't always work in the micrometastatic setting, so we have to be careful. True biomarkers are very hard to find. I wonder if all those different trials could pool their molecular data, and maybe we could learn something to plan the next ones better as well. Okay, so now I'm going to focus on the very busy last 5 years and ask some of these questions. First, success of genomic-driven trials. So we had the Clarity trial with IDH1 meeting its primary endpoint of PFS with crossover in a very ethically designed trial. This is certainly a pivotal biomarker selected population in a rare cancer, so really hard to do. Then we went into this era of FGFR inhibitors, and there were many drugs. Pemigatinib was the first FDA-approved drug for the indication of cholangiocarcinoma in history. I always remember looking at this and thinking, wow, just about everybody with this fusion gets some benefit, and we know that a small bit of shrinkage can make someone feel a lot better. These have now moved into the first-line setting. And then there were these beautiful studies that came out, this one by Lipika Goyal, where they followed what happened to patients who responded to an FGFR inhibitor and then developed resistance, and beautiful documentation of these polyclonal secondary mutations that happened at the time the tumor took off, which led us now to next generation FGFR inhibitors like futibatinib, this nice phase 1 trial by Funda that showed that this drug had activity against a broader range of FGFR aberrations, including patients who had seen previous drugs and had developed resistance. And it went on to phase 2, lovely paper in the New England Journal, objective response rates of 40%, durable PFS, and a toxicity profile that's a bit of a handful but can be managed. And then the next kid, Relay 4008, a lot of excitement here. This drug seemed to have real activity preclinically in models where other drugs failed, seemed to have activity in patients where there were resistant mutations developed, and it seems to have a high activity in patients who are seeing FGFR for the first time, although this needs to mature and see how durable these responses are. Now, changing targets, cholangio got a lot of help from agnostic trials, and so targeting BRAF or V600E on the ROAR trial, You can see again, most patients with this mutation benefiting from treatment with davrafenib and trametinib. And then even very rare things, these NRG fusion with this new drug, zenocutuzumab, and you can see in yellow a couple of patients there responding nicely in these rare mutations. And then, of course, there's been so much advancement in targeting HER2, and that's exciting for us because we know that these amplifications and overexpression are much more common in the extrahepatics and the gallbladders that really need some help. And I've listed here a number of HER2-directed therapies that have been evaluated that look very promising. I'm quite excited about the bispecific HER2-targeted antibody, zanidatamab, with 47% response rate in cholangio in phase 1. So that number line now has gotten quite busy. You can see I've also added an NTRK there, and you can see these advances coming in with the genomically driven trials. So real poster child of success here. I'm optimistic, so I've added in gray the HER2 and NRG. Maybe we'll see that in 2023. And I put this figure up just to remind me to make the point that CCF has made so well and others have made. There's really no excuse today not to get a tumor or a liquid biopsy routinely in patients because almost 50% of patients may be treatable with something targeted, whether it's on an approved agent or on trial or off-label. We need to be always thinking about this. We also have to better define the clinical responders and get more tissue to understand resistance. And what's very exciting is that if you make a table in the last month of trials, you'll see that most of these are biomarker-driven trials now in cholangiocarcinoma, and they're not all the same targets we've been talking about. There's DDR there, and there's other ones, and there's going to be tons of new data coming. So now I'm going to finish off the last 3 questions. What could we use in the second line after GEM-CIS? What about dose intensity chemotherapy in first line? And finally, do we see a benefit of immunotherapy? So ABC06, Angela's over there, yes. A nice trial that showed the benefit of FOLFOX versus best supportive care after GEM-CIS. Modest benefit, but a hazard ratio of 0.69 can't be ignored. So we have that standard while others develop. The chemotherapy dose intensity, never a dull moment in the first-line setting. 4 trials, only 1 positive. In Japan, adding S1 to GEM-CIS is modestly a positive trial, probably effective there. We know this drug's not available in the West. The French, Prodige, tested FOLFIRINOX. It works in pancreas, so surely it works in biliary cancer. Negative trial, numerically favoring GEM-CIS, and I think the failure can't be explained by dose intensity or toxicity, so it's puzzling. Disappointing, SWOG 1815, Rashna's over there. The very promising GAP regimen with nab-paclitaxel added to GEM-CIS also didn't meet its primary endpoint, although there are some signals in gallbladder and locally advanced that I think will be studied further and this regimen may become useful in select populations. And then finally, Nutide 121, which is really a dose intensification of gemcitabine to make it more available in cancer cells with cisplatin versus GEM-CIS. Closed at the interim analysis for crossing futility, and we'll present this later this year. So finally, positive, TOPAZ-1, adding the checkpoint inhibitor durvalumab to standard of care GEM-CIS, and you can see the curves separating for survival and PFS, although a hazard ratio of only 0.8. With a little bit more time, the curves looked a little bit better with a hazard ratio of 0.76, and everyone talks about the landmark of 2 years where there's more than a doubling of survival, which is something meaningful and something you can talk to patients about. This data was just recently presented by Dr. Vallée and colleagues at the Asian ESMO, and to me, it's exciting to see a large global trial present next-generation sequencing data. They had 64%, or 440 patients, with data, which is well balanced between the two arms. And so there you are, you have a good glance of what does an all-comers global genomic landscape look like. And you can see the rarer actionable ones in light green. I'm ever the optimist. I changed KRAS there at 24% into sort of a medium color because I think it might be coming. But you can see a lot of the main drivers are not yet druggable, like P53. And then when they looked at the forest plot of this across the different subgroups, fairly reassuring signal that there really doesn't seem to be a lack of benefit of the topaz across those different subtypes, although there are some wide margins. We heard the press release that Keynote 966, which is asking pembrolizumab added to GEMSIS as a positive trial. This is a little different from TOPAZ in that it is a bigger trial, and it allows chemotherapy to continue the gemcitabine arm to continue past 8 cycles, which is a little bit how we practice here. So we look forward to seeing that. So there's my timeline. I did it myself, so it's not quite as elegant. But I think what you can see is it has been an extremely exciting time for biliary tract cancer. You can see in black all the genomically driven advances that are now come into practice. In blue, you can see the chemotherapy arms that are stalwarts in GI oncology remain important in patient care even today. And then in purple, you can see the impact now of immunotherapy. And I'm optimistic for new drugs in 2023. So to finish, progress in 10 years. So biliary tract cancer, I think we can say firmly has entered the era of precision medicine and immuno-oncology. We still have many problems to solve. We need biomarkers for selection and resistant pathways, a lot of science going on. We have to optimize these therapies now, sequencing and combinations. We have to continue to improve access to profiling and precision drugs in routine care, not just at the big centers where we know to do this. And we have to further improve the patient's journey in this rare cancer. Now, CCF has moved that needle remarkably in 10 years, but we have a ways to go. I see lots of patients come to me who've had no profiling and didn't know. What we have seen is accelerated drug approvals in a rare tumor. I don't remember seeing that 10 years ago, and CCF was there to help with that, based on phase 2 data, non-randomized data, tumor-agnostic approvals. And so now we're seeing more novel designs and targeted frontline therapies, which is very exciting. Now, we have numerous studies. I don't have time to go through them, but a lot underway to try to treat more patients with curative intent using multimodality strategies, perioperative treatments, downstaging, transplant, radiotherapy, all of these coming in, localized therapies. And to get these done, we'll need more international collaborations. And I haven't given any data, but I do think that we all share this excitement that there is a promise coming of earlier diagnosis with platforms of circulating tumor DNA and artificial intelligence that may very soon start to identify solid tumors, difficult ones like this one, much earlier, where we can do more. So I would like to say thank you to the Cholangiocarcinoma Foundation. It's been a real pleasure for me to be involved over the years and gave me a place to ground and talk to other people who knew about this cancer. And I thank you for listening. Bye.